in bacteria?
Sir, transformations?
Okay, just wait. Okay. Yes, my voice is audible.
Yes, sir.
Yes, sir.
So, and what are the sexual reproduction in bacteria?
Sir, conjugation. Transformation.
Conjugate. First of all,
Transduction.
transformation. And who is discovered by the transformation? The scientist Griffith. He is conducted
Sir,
one experiment.
Griffith.
Like he taken some virulent 6 batches of mice, and the bacteria. What is the bacteria? He taken pneumonia, right?
Yes, sir.
So on the transformation also, what happening? The exchange of genetic material. Next one is the conjugation. The conjugation also cell to cell attached through the conjugation to the genetic material, one cell to another cell, another bacterial cell, and transduction. Okay, these are the some bacterial reproduction. So coming to viruses. So in viruses, we are not using reproduction. We are using replication. What is the meaning of replication? Replication.
Making the copy of itself.
Yes. Simply, the replication definition is the process of multiplication. It is called as replication, right? The process of multiplication. So, the replication, the process of multiplication is viruses is generally referred as the replication. A replication of viruses takes place in the 2 different ways. So, first one is the lytic cycle. Second one is the lysogenic cycle, right? Lytic and lysogenic cycle. How the way, what are the steps are involved in the lytic cycle? How the lysogenic cycle steps involved, we will discuss. So, generally, the lytic cycle, it is also called as growth cycle. Lytic cycle, it is also called as growth cycle. One step growth experiments is conducted by the scientist Delbruck and his co-workers. His co-workers enable to understand the viral growth cycle. This, in detail, they are explained. My voice is audible?
Yes, sir.
Okay. So, the lytic cycle, it is also called as the growth cycle. This one-step growth. The one-step growth experiments done by the scientist name is Delbruck and his co-workers. Detailed explain the scientist. So before that, some of the terminology is there. So some of the terminologies, generally all the bacteriophages exist as a vegetative phase in the host cell for some time, right? But in some phases, the viral DNA gets incorporated with the bacterial chromosome, which are known as prophase. Prophases. These prophase able to become prophases are called temperate phases. And those unable to do are known as virulent phases. And the multiplication process of virulent phases is referred as the lytic cycle. And whereas the temperate phases is known as lysogenic cycle. These are the definition of lytic cycle or lysogenic cycle. So what are the steps are there in the lytic or growth cycle? T-even bacteriophages exhibit lytic cycle. Already, what are the nature of like bacteriophages? It is always in the T-even numbers like T2, T4, T6, T8. Okay. T-even bacteria, only the T-even bacteriophages exhibit the lytic cycle. So the T-even bacteriophages multiply through this cycle, through the lytic cycle. These all are the bacteriophages in the even numbers. T2, T4, T6, T8, 10, 12, 14, whatever may be. So these viruses infect the bacteria. Generally, what is the bacteriophage?
The virus that infect bacteria.
The virus affect or attack to the bacteria or infect the bacteria. That is called as bacteriophages. Same like this, the virus infect the bacteria, degrade their DNA and kill the host cell, rupturing the cell wall. It is simply, the definition is, it is known as lytic cycle. This lytic cycle, the ways are, different ways are there. In the next slide I will explain. But the definition of lytic cycle. The viruses, when viruses infect the bacteria, infect the bacteria, then what happening? Degrade their DNA and kill the host cell, rupturing the cell wall. This entire process called as the lytic cycle, right? So these phases kill the host cells. These phases are called virulent phases. When enter to the virus, infect the bacteria, degrade their DNA, and then kill the host cells. These phases, this type of phases are called virulent phases. Right? Do you understand?
Yes, sir.
What is the lytic cycle? When, very simple, the virus attack to the bacteria or infect the bacteria, what happening? Degrade, once attack or once infect the bacteria, the bacterial DNA is degraded, kill the host cell, rupturing the cell wall. This process, the rupturing the cells, they are involved some enzymes also. So that is, we will detail in the next slide. So this entire process known as lytic cycle. These phases kill the host cells. These phases are called virulent phases. What is the meaning of virulent?
Lethal.
Pathogenic, sir. Pathogenic.
Virulent. Pathogenic,
sir.
Okay. So these are the lytic cycle or growth cycle. The 4 steps or 4 ways or 4 phases are involved in this, the lytic cycle. First one is the adsorption. Second one is the penetration. Third one is the latent period. The fourth one is the lysis. Okay, do you understand? How many phases are there in the lytic cycle? How
4,
many
sir.
phases are there in growth cycle?
4, sir.
4.
4, sir.
First one is,
Absorption.
second one is,
Penetration.
so third one is,
Latent period.
Latent period.
the fourth one is
Fourth
the
one is
lysis.
lysis.
So what is the meaning of,
Lysis.
general meaning of absorption?
Attachment, sir. Attachment on the surface of the host.
Okay, attachment. So penetration, any idea?
Injection, sir. Injection.
Okay.
Injection.
Latent stage, any idea?
Latent period means in the after going into the host cell, the viral cells can multiply their number.
So like maturation phase, it will take some time.
Maturation phase.
Okay, the final one is the
Lysis.
Lysis means degradation of the host cells are coming out of the host cell.
Yes, the breaking breaking of the host cell. Automatically they release the virulence. These are the simple lytic or growth cycle. So the first one is the adsorption. Okay. So the absorption is attachment of the virion. What is the virion? Why? Yes, yesterday we discussed, I think. Yes, tell me.
Sir, can you repeat the question, please?
What is the virions? Already we discussed yesterday virions, prions.
Virus particles, sir.
Yes. The small virus particles are called
Virions.
Yes.
Virion, sir.
So, the absorption is the first step of the lytic cycle or first step of the growth cycle. The attachment of the virion. Virion, it is nothing but the virus particle, right?
Virus particles.
But the small virus particles, what we called in the virology nomenclature, virions and prions. Yesterday we talked about the virions and prions. So the attachment of the virus particle to the host bacterium is called adsorption, right? What is the meaning of adsorption? Attachment of viral
Virions to the bacterium.
virus particle to the host bacterium. The virus, where it is in the lives in, it is the bacterium. Bacterium is the host, right? In case the virus, it is also host as a plant. The bacteria, sometimes the plants act as a host. Sometimes human beings also host. Sometimes animals also. But this lytic cycle, this stage only occurs in the T-event bacteriophages, right? You understand? Yes?
Yes, sir.
Yes, sir.
Do you understand?
Yes, sir.
Generally, the virus causing disease human beings, the virus causes disease in the plants, the virus causes disease in the animals, the virus causes disease in the algae members, the virus causes disease in the bacteria, the virus causes in the, already we discussed in the previous class. So the virus compulsory, the virus needs to some host, whatever that is bacteria or fungi or animals or human beings or plants, whatever may be. But here the lytic cycle or growth cycle, it is occurs only in the bacteriophages, right? So that's why the virus, the attachment of the viral particle only, it is attached to the bacterium. Bacterium act as a host. This process called as adsorption. Very simple. The attachment of the virus particle in the host bacterium is called adsorption. Right? Clear?
Yes, sir.
Yes, sir.
So this involves in the physical mechanism of collision between the virions and the host cells. The virions attach to the host cell wall with the help of the tail fibers. So how virions means it is the small particles of viruses. The small particles how to attach in the host bacterial cell wall. In this Already yesterday we discussed in the structure of the functions of the bacteriophages. The bacteriophages, two major parts are there. One is the head part, it is hexagonal pyramid-like, and in the lower part is the tail part.
Tail part.
Tail parts is tail pins are there. Tail
Fibers are
fibres are there. Okay. So
yes, sir.
in this, the virus particles attach to the host bacterium cell wall with the help of the tail fibers. These are the adsorption process. First of all, very simple. What is the definition of adsorption? The simple
Attachment of the
attachment of the
virions
Virions to the
virus
bacterium.
particles,
The
the host
bacterium.
bacterium is called as the adsorption.
Adsorption.
How it is involves in the, how attached is the, it is a physical mechanism of collisions between the virions and the host cells. So, and with the help of tail fibers. Okay, these are the regarding absorption. And the next one, the second is the penetration. The slide is like absorption, not absorption, adsorption. After adsorption, the bacteriophages are able to release an enzyme called isozyme. What is the enzyme? The penetration process. The bacteriophage are able to release an enzyme called lysozyme. What is the role of lysozyme?
Lysozyme.
Hydrolysis of the bacterial cell wall. What is the meaning of hydrolysis?
I think it will be dissolved the host cell wall, sir.
Yes, that is the hydro. Hydro and water. Lysis means break.
Breaking of water molecules, sir.
So here, the first step, completion of the adsorption, the bacteriophages are released to the enzyme. The enzyme name is lysozyme, which is the function of this enzyme hydrolyzes the bacterial cell wall. And this enzyme system is supposed to drill a hole in the bacterial cell wall through which nucleic acid is injected to the cell. Do you understand this sentence? This enzyme system is supposed to drill a hole in the, how to give injection, doctors or nurses directly. First of all, take the needle, syringe, then how to give directly the left side of tissues or whatever may be. How they give? They drill the hole.
They'll poke a hole.
But directly the nurses directly given to our tissues. So that is the process is drill. Here, this enzyme, the bacterium host cells, this enzyme, lysozyme enzyme, it is what is the role of this enzyme? It is drill a hole in the bacterial cell wall through which nucleic acid it injected into the cell. What is the nucleic acid means?
DNA or RNA, sir?
Yes.
Genetic material.
It's genetic material.
DNA or RNA. So this enzyme entered to the bacterium host cell. This enzyme started a drill of hole. Then after holing, which nucleic acid injected into the bacterial cell. So already the virus genetic material, DNA or RNA, whatever may be. Sometimes DNA present, sometimes RNA present. Most of the plant viruses contains RNA only. Sometimes it is depend upon the virus nature. So immediately after drilling up the hole, what we can, the injection, some medicine, the nurses or doctors, they are giving the injection. First of all, they given to the injection through the some medicines. So here, same like this virus, when attached to the bacterium cell, after drilling of a hole, they give the nucleic acid. The nucleic acid, it is nothing but either DNA or RNA inject to the bacterial cell, right? The injection of the phase nucleic acid into the host cell is known as the penetration, right? Do you understand this penetration? Simply,
Yes, sir.
so the bacteria phase means it is nothing but virus, but the virus, it can be infect the bacteria, what we call bacteriophage. The bacteriophage able to release the enzyme. The enzyme name is lysozyme. The lysozyme capacity or the lysozyme role is drill a hole. After drilling a hole, this enzyme release the nucleic acid. The release, it is nothing but injected to the host cell, right? The injection of the phase nucleic acid into the host cell is known as penetration, right? Do you understand this penetration step?
Yes, sir.
So the protein shell remains outside of the host cell, which is referred as the
Ghost.
So the contraction of the sheath facilities of the injection of the DNA into the cell, the phage DNA injection, the host DNA degrades and the cell is forced to make viral constraints. Do you understand this sentence? First of all, after the virus given the viral nucleic acid to the bacterium host cells, what happening? Once enter to the viral genetic material into the bacterium host cell, the bacteriophage, the bacterial DNA is degraded, right? After degrades, it make viral constraints. Do you understand this sentence?
Yes, sir.
The bacteria naturally consist DNA material. Whence the viral, after injection of these nucleic acid, these DNA RNA, the bacterial original DNA material is breakdown. Then this viral DNA or RNA, it becomes a viral constraints. This is the penetration step, right? Do you understand? Yes? Any doubt?
Yes, sir.
So next, third period is latent stage, right? This is divided into two phases: eclipse phase and maturation phase. What is the third phase is? Latent phase. It is divided into two phases: eclipse phase and maturation phase, right? So in eclipse phase, the virions are not detectable. In maturation phase, parts of the new virions are synthesized separately by using host metabolic system. So after maturation phase, they shows head, tail plate, and tail fibrae. Later, all the parts are assembled to form a complete virions. Virions means complete virus particles, right? Yes, please respond.
Yes, sir.
Latent period, very simple. How many stages in the latent phase? Two phases. Eclipse phase. What is the what happening in the eclipse phase? Yes, tell me.
Virions are not detectable.
Yes, virions are not detectable in the eclipse phase. So what happening in the maturation phase?
New
Synthesized
virions are synthesized separately by using host metabolic system.
So in this, the maturation phase, the virions are detectable. The eclipse phase, the virions are not detectable. The maturation phase, the virions are detectable. What are the parts are detectable?
Sir, that means, sir, detectable mean?
Detectable. Detectable means it is appears. Not detectable means the eclipse phase, there is no virion parts at all in the. So when start the latent phase, in the first page, sometimes they're not appearing any parts of the like head, tail plate, tail fiber. There is no appears of the parts. That phase, eclipse phase. So that's why we are using the virions are
Detectable.
not detectable in the eclipse phase. In the maturation phase, after some time, the maturation phase, there after synthesis of these virions, the appears is the head, tail plate, and tail fibers. These are the maturation phase. These are the detectable. Eclipse phase is the not detectable. Okay. After all the parts are assembled to form the complete virions. Right? Do you understand? Yes.
Yes, sir.
Yes, sir.
So this is the latent period. Next, lysis. It is very simple. Yes. All of you know lysis. Simply lysis. Lysis means it is breakdown, right?
Yes, sir.
At the end of the latent period, the host cell wall ruptures and all the virions are released. Simply, the lysis means it is rupturing or destroying or breakdown, whatever may be. In the host cells, what happening already the latent maturation period, there all the parts are assembled to form the new virions. They are waiting for the release. Then lysis. The lysis process, the host cell wall ruptures
And releasing of
breakdown.
the.
So what happening? The virions are released. These are the lysis process. This known as lysis. The cause of lysis is assumed to be due to the presence of the again lysozyme enzyme. It is capable of the dissolving the cell wall. Okay, the enzyme in the entire this cycle, the bacteriophage are able to release the enzyme. The enzyme name is lysozyme. It is very useful enzyme in this cycle. Okay, finally the lysozyme dissolve the cell wall, then automatically the virions are released. The average number of virions is specific for a host virion system are referred as the burst size. Okay, these are no need in your exam. These points are the enough for your exam. In this and your exams, either it is long answer. They are the question paper. Sometimes they are asking long question. Replication of viruses. You write lytic cycle and lysogenic time. Sometimes they are only asking short answers, either lysogenic or lytic cycle. Sometimes they are asking only growth cycle of virus. The growth cycle is nothing but the lytic
cycle.
Okay. So these are the these are the outline of diagrams and different steps. Absorption, how enter to the virus in the bacteria. Okay, this is the bacterial cell, that is the phase. Okay, these are the phase DNA and the protein code. It is what we call in the ghost. So these are the eclipse phase. You observe the eclipse phase and maturation phase. What is the two different images are there? In the maturation phase, we directly we see, oh, these some like bacteriophages. Bacteriophages means some viral particles. In the eclipse phase, like thread-like structures are there. In the right side maturation
Only the
phase,
nucleic acid.
some structure are there, like
Tail-like structures are there.
head. So that is the eclipse phase. So that's why we are not detectable in the eclipse phase, and detectable it is the maturation phase. Now finally, the lysozyme enzyme, it is capable of dissolving the cell wall, break down the cell wall, then release the
Variants.
yes. And this is the lytic cycle or growth cycle.
Growth cycle.
Okay.
Yes, sir.
So do you understand?
Yes, sir.
How many steps are there on the lytic cycle?
4, sir.
3, sir.
Four.
4.
Absorption,
Penetration,
penetration,
Latent phase and lysis.
latent,
phase, and lysis. Very simple. Just you learn this diagram. It is simply right. Only what are the main things? The absorption, attachment of the virions to the host cells,
to the bacteria.
and next one is the penetration.
Penetration.
Injection.
Lysozyme.
After attachment of the virus, generally the nature of bacteria phase, they are release the lysozyme enzyme. The lysozyme enzyme, it is drill the hole and like injection type. When the virus give to the injection to the bacteria, what happening? The viral DNA went to the bacterial cell. So that is called as phase DNA. So then what happening? After penetration, what is the latent stage? The latent stage, in the two helix phase and maturation phases are there. In the first of all, beginning time, there is no detectable phase called as the eclipse phase. And then go for the maturation phase. The maturation phase, some like tails, fibers, fibrary plate, these are the detectable. What we call in the maturation phase. After lysis, again here the lysozyme enzyme, it is capable of dissolving the cell wall. Once dissolving the cell wall, automatically released the new virions. These are the regarding about growth cycle of viruses. So next, coming to lysogenic cycle. Yes, lysogenic cycle. So the lysogenic cycle, it is a link between the lytic cycle. Okay. Yes. As mentioned in the earlier, there is another kind of the multiplication process known as the lysogenic cycle. Very simple. In this, the first lytic, what happening in the lytic cycle? The finally, what we find out in the final
new virions.
New virions. So it is single to many virions, right? So that is the process is the multiplication process, right?
Okay.
This in this cycle also, the finally what we finally found out, multiplication of the virions, multiplication of the new viruses. This process is known as lysogenic cycle. So same in the lytic cycle, lysogenic cycle, finally what happening? Multiplication is the
virus.
Yes, multiplication is the very, very important. But here in this bacteria cell also, the reproduction is, that is same process. What are the findings of the bacterial reproduction? The exchange of genetic material, but different ways here, like transformation, like conjugation, like transduction. Okay. But what we final exchange of genetic material. Here, the two ways are there, two cycles are there. The two cycles, the process is different, but finally
multiplication of virus.
Multiplication process, right?
Yes, sir.
So here lysogenic cycle also, multiplication process is known as the lysogenic cycle. In this process, the phage infects the susceptible cell without causing immediate lysis. Without causing immediate lysis. In this cycle, the bacterial DNA is not destroyed, but phase DNA is incorporated with it. Like the phage DNA gets attached with the bacterial DNA and replicates along with it. So do you understand this sentence in this cycle? So what happening in the lytic cycle? When enter to the viral DNA enter to the bacterial cell, the bacterial DNA is destroyed. But in the lysogenic cycle, the bacterial DNA is not destroyed. But what happening?
Good morning,
Phase DNA means? Phase DNA
Phase DNA, viral
it
DNA,
is
sir.
nothing but virus DNA.
The
Right?
viral DNA is get incorporated in the bacterial
Incorporated with it.
chromosomes.
So the phage DNA gets attached with the bacterial DNA. Do you understand? There is no destroying the bacterial DNA. What happening here? The phage DNA gets attached with the bacterial DNA. When this viral DNA enter to the bacteria in lytic cycle, when enter to the viral DNA into the bacterial cells, immediately the bacterial DNA destroyed. But here there is no destroying. What happening? It gets the attached with the bacterial, the phage DNA and bacterial DNA attached. Then replicates along with it. Do you understand? It is very simple. There is no destroying. They are combined. They are replicates. Combinedly replicates. Right?
Yes.
Yes, please respond. Do you understand? Once again, I repeat.
Yes, sir.
Yes, all of you, please respond.
Yes, sir.
Yes, sir.
I got some messages. Okay. Okay. So, in this process, the phage infect the suspectable cell without causing any immediate lysis. So, already repeated. The viral genetic material becomes an integral part of the bacterial cell. What happening? There bacterial DNA and viral DNA attached along with replicates. What happening? The genetic material also becomes the part of the bacterial cell. So, do you understand this line, this sentence?
So, does that mean that this viral genetic material not causing any harm to the bacterial cell?
Yes. This is the point. But it is together. Both commands, they are replicated. The both command replicates. So, what happening? The genetic material becomes the part of the bacterial cell also, no?
Yes, sir. But does it cause infection or does it not cause infection?
No. This is the process of how once enter to the bacterial cell. These cycles only in the bacteria. Okay.
Yes, sir.
So, but in the lysogenic cycle, there is no harmful.
But ultimately, it is killing the bacteria there and releasing the viruses,
yes,
right,
yes,
sir?
Finally.
What about here?
Just wait.
Yes, sir.
Okay. There is no, again here. So here, there is no lysis phase. So there is no rupturing of lysis. They are together release the, suppose, for example, in the lytic cycle, in the last phases, the lysozyme enzyme break down the some cell wall. Here, there is no breakage of any lysis phase. Directly, they are combined with to develop the new viral particles. In the first step, you see the diagram, attachment of the virions to the bacterial cell, then entry of the viral DNA into the bacterial cytoplasm. It is a viral DNA and bacterial chromosome. This one, division of the some bacterium. So here there is no harmful of this lysogenic cycle. Okay. Yes, please respond.
Sir, after completion of the lysogenic cycle, it will follow the lytic cycle, sir, or it will be remain like a lysogenic only?
Lysogenic only.
Okay. It will not follow the lytic cycle again.
Yes.
Okay.
That means there is no rupturing of the bacterial cell,
Yes,
but
yes,
it
that.
remains like that.
Yes.
Okay, sir. Thank you, sir.
Okay. So these are the very simple. So these are the some difference between the. Just wait. Okay. So lytic cycle and lysogenic cycle. The viral particle multiplies in the host. The only viral genetic material replicates in the host. So this is the difference. You ask question. Na? The first one is the. So the host cell is damaged. The host cell is not damaged immediately. So prophages are not found. In this, prophages are formed. Those viruses which perform this cycle are called virulent phases. The lysogenic cycle is performed in the temperate or virulent phases. Host DNA is damaged. In this, host metabolism is not controlled. T-even phases, bacteriophage. The example of a lysogenic cycle is very important. It is the like lysogen, polyphase lambda. That is not polyphase 1. That is lambda, polyphase lambda, example of the lysogenic cycle. Right? Yes.
Yes. Okay, sir.
So next.
Sir, sorry for the interruption. Sir.
Yes.
Sir, then what is the example for lytic cycle, sir? Is it T2 bacteria?
That is some T-even phases, bacteriophages. Hello. Yeah. Yes, ma. T-even phases.
Sir, your voice is breaking, sir.
T-even phages, like the lytic cycle only.
T4.
Yes. Here, general some phage bacteria, but this is different. Not not T-even bacteria. Any phage bacteria. The virus attack infect to the bacteria. That is even and odd numbers are there, but the lytic cycle only T-even phages. But here there is no T-even, any bacteria. So okay, coming to diseases caused by the virus. Already you are studied in degree level, I think intermediate level. So they are the very simple. So plant diseases. The plant disease caused by the viruses.
Mosaic disease.
Yes, first one is the some important, many diseases are some important according to our syllabus, and some mostly we are regularly appear in the viral diseases in the plants. So first one is the chlorosis. What is the meaning of chlorosis?
Yellowing, sir. Yellowing coloring, yellow coloring
Yes.
of leaves.
The loss of chlorophyll, sir.
Yes,
The loss of chlorophyll.
Automatically loss of chlorophyll. Automatically the leaf
turns to yellow color.
Chlorophyll catches
entirely due to the loss of mainly chloro. Chloro means the loss of chlorophyll. Okay. So automatically the plant leaves are. Please mute yourself. Yes.
Renuka, please mute.
For example, you observed in the rice yellowing diseases. Anyone farmer background? Anyone once you observe the field, paddy fields. Paddy fields, these chlorosis mostly seen in the rice fields. So next, coming to mosaic. Yes. Mosaic means it is also color.
So light and dark spots may be seen on the leaves.
It is a color change. The color whatever may be. So here also some in any disease, first of all, the loss of chlorophyll, they are not able to synthesize the food material automatically. They are not, the entire life cycle is stopped. Okay. Without photosynthesis, the plants not can survive. They're not doing the photosynthesis process. Okay. So the mosaic also, it is a color change that occurs in the isolated patches in the green lamina. The mostly in the mosaic virus, mosaic means in how to tell,
Mottles,
leaf curl, like leaf curl, the back side and the front side. Leaf curl also, leaf curl. So it's a murchukunta idea on the once enter to the any viral disease. So the leaf is like
it's curling, sir. Upward
yes,
curling,
too much of
downward
curling, too much of curling. Like the leaf is round shaped also. The leaves, all the leaves are the round shaped, and after some days fall down the leaf, entire leaf. But the mosaic effect is in the color change of the leaf surface, and mostly affected part is the lamina. You know lamina, the center part.
Yes, sir. Which gives rise to
flat
veins
parts,
and
sir.
veinlets.
Yes, which, okay. Vein clearing.
Vein clearing. Yes, sir.
This is also related to that. It is the symptom where is the tissue close with the veins turn into the yellow. The remaining areas remaining greens. So, you know, some vein parts only change the color, loss of color. The remaining in the vein to vein, some places are there, leaf places, that place is green, but the vein clearing. So, what happening?
Veins
The day by day, the veins are, entire veins are is destroying by the viruses. Automatically, the leaf also fall down. So here it is also the same problem. Okay. So the vein clearing, automatically the veins are becoming the yellow color. So generally these symptoms mostly seen in the bendy vein clearing. Bendy, you know,
yes, sir.
bendy,
Yes,
lady
sir.
finger.
Yes, sir.
So vein bonding. This condition, tissue close to the vein remaining green, same happen. It is the lamina undergo chlorosis. So this disease only mostly seen in the citrus vein bonding. Next, ring spots. You know ring spots?
Yes, sir. Papaya ring spots. Ring spots, papaya leaf లో papaya పైన కనిపిస్తాయి సార్ బాగా.
Yes, the loss of chlorophyll in the circular spots, circular round parts. That's why we called ring spots. The ring spots due to the viruses. The ring spots mostly seen in the tomato spotted wilt. So yellow color spots you observe when you are growing in the tomatoes in like terrace and surroundings your places. Just you observe the yellow color spots. That is the ring spots of the that is the viral diseases caused by the some viruses. Okay, next, dwarfing or stunting. What is the meaning of dwarfing and stunting?
Stunting means
Reduce
plant
the
becoming
growth of
short,
the
sir.
plant parts. Yes, yes. It is reduced the growth of the plant parts, right? This one shows like rice fields also. The dwarfing and stunting, it is reduce the growth of the plant parts. It is rice stunting. Okay. So next one is the malformation. You know malformation? Anyone idea? Mal.
No, sir.
It is abnormal growth of the plant. Abnormal growth. Either abnormal growth, either entire plant, either plant parts also. That is the malformation. So it is some I forget it, but the malformation, some coke of okay, I forget it as example. So breaking of flowers. So breaking of flowers, the loss of color in the petals in certain parts and formation of the dark colors and other parts also. So this is example, the tulip mosaic breaks. Example is tulip mosaic diseases. It is caused by the viruses. So next one is the necrosis. You know necrosis? What is the meaning of necrosis?
నెక్రోసిస్ అంటే షూట్ గానీ అది డ్రై అయిపోయి అసలు అక్కడ
Becomes
డెత్ వస్తుంది సార్, సెల్ డెత్, సెల్స్ డెత్ అయిపోతాయి సార్.
So many viruses attack to their host cells and rapidly kill them. It is immediately. It is a very dangerous disease of necrosis. Okay. The many viruses attack their host cells and rapidly kill them. This is killing is called necrosis. The necrotic tissue is demarcated from the living tissue by the distinct brown border. It is mostly the this necrosis in the tobacco necrosis and potato ring spots. These are the examples of necrosis. Okay, this is the some general many diseases are there. So we are botany. That's why we coded plant disease. But you are aware about compulsory what are the bacteria diseases, what are the virus diseases. Okay, the plant diseases caused by the viruses, the plant disease caused by the bacteria, compulsory. And also same time same what are the animal diseases caused by the bacteria, caused by the viruses. Okay, humans, what are the bacterial diseases, what are the viral diseases, compulsory some idea. So according to the some symptoms, oh this is belongs to, I think this is the bacterial disease. Oh, I think to compare to the bacterial and virus, the bacteria, most of the bacterial diseases it is curable, but the viral diseases not curable because it is very dangerous, right? You know, like corona. Many HIV, AIDS, and HBU, many diseases. The viral diseases very, very dangerous to compare to the bacterial diseases. Yes. So, but you aware about not only plants, animals, human beings. Okay. What are the viral diseases? What are the bacterial diseases? Compulsory. Okay. You learn. Next. These are the already I shared notes. Next, transmission of viral diseases. Transmission means spreading. How to spread? Different ways of different transmissions are there. Okay. So the mode of transmission of viruses and diseases caused by them according to the virus and the host. Here, the transmission of viral diseases, the host is compulsory. So the bacterial disease, there is no, it is compulsory host or not? So the viral disease compulsory host inside of the host. In the first class, we will discuss in the before day, we will discuss. It is the obligate parasites and inside of the host. Okay. They are the virus living in the inside of the host cell. Okay. So in very few viruses are freely transmitted through the air, water, and contact. Okay. So remaining things, some any idea? These are the regular process. Transmitted air, water, and contact. So what about others? The majority of the viruses transmitted from the one host to another through the like plants, vegetative parts, and cells of fungi and phanerogamic plant parasites. Okay. The general is air, water, and through contact. So majority is one host to another through another host. One host to another through the vegetative parts. Cells up. Suppose pollen grains are there. Okay. Pollen grains. Some bees are collected some cells have to transfer to the another plant. So that time also the viral diseases transmitted through insects also. Okay. So, and in the majority of transmitted in the vegetative parts. What are the vegetative parts? In vegetatively propagated plants like potatoes, sugarcane, banana, rose, these concerned viruses are transmitted through the buds, stem cuttings. So here, suppose for example, sugarcane. How to cultivate sugarcane? Anyone knows? Yes.
Through stems, sir.
Through stem. How? The stem cuttings.
Stem cuttings,
Nod,
yes.
nod దగ్గర
The
కట్
nod.
చేస్తారు.
Yes,
Nod.
yes.
Nodes, sir.
So this is a very major transmitted some viral diseases. And potato. How to cultivation potato? This is also cutting up the potato ice.
Potato
like
ice, sir.
ice part. So they cut the ice part, then plantation. So sometimes some buds also. Some are transmitted to the grafting. You know grafting. It is everyone knows grafting. So
Yes, sir.
So and one another plants cut the and then so you grafting method. You know grafting method. So then tight and sometime leaving the some for few days automatically exchange of the some through the water and nutrition through the xylem phloem it is enter to the other plant. This is the grafting method. You know grafting method?
Yes, sir.
So next one, mechanical transmission. Mechanical transmission, the virus like potato virus are transmitted through the sap of the infected plants. This type of transmission usually takes place through the physical contact, agriculture implements, and hands of the labor. So when will cultivation of potatoes, suppose already the potato got some viral diseases, then the laborers cut into the pieces, then the plantation is planting these potato. That time also it is very. Otherwise, already the labor also got some viral diseases. The viral diseases wounds and something. So whatever may be. So automatically it is easily through the transmitted through the man to plants to soil. Okay. So most of the mechanical transmission we are seen in the agriculture. Okay.
Yes, sir.
The seed transmission. So here it is very, very important and very dangerous. First of all, these are the reason. Most of the farmers, not most, 90% of the farmers, they are going to cultivation any plants like paddy, cotton, chili, whatever may be. Most of the 95% of the farmers, they are not using the seed treatments. They are directly selling the shops, then go to the directly cultivation of the plants. There is no awareness, there is no idea. But this is the very, very big problem in on our agriculture farmers. They are not doing any seed treatments. Okay. More than 50 plant viruses are reported to be transmitted through the seeds only.
Seed transmission.
Many diseases through the seed transmission.
Okay.
Many diseases. So like for example, bean mosaic and the lettuce mosaic, any viral diseases. Most of the highest. Please mute yourself, please. Yes. Anyone please mute yourself. Who is talking? Just wait, I will.
Uma Maheshwari, sir.
Uma, please mute. Sir, you can mute it from your end, sir.
Yes. Done. Okay. So most of the viral diseases, plant viral diseases are highest reported through the seed transmission only. Okay. So already I discussed pollen grains. The pollen grains produced from virus-infected plants usually possess the viruses. Such pollen grains transmitted the virus in the pollinated plants. This is the main base is the seed is the base, right? Once the seed done seed treatment, it is becomes a healthy plant. It is very good, but most 90% of the farmers not seed treatment, okay, directly. Some of in the old days in the villages, they are not using. Already suppose this year seeds are available, they are some shade dry and some drying, then same that seeds are used in the next year cultivation. So in this period, so many virus attack to the like we air in the different contact to the seeds, touch the seeds. So they are not going for the seed treatment. So because of the most of the like cotton, like chili and turmeric, these are the reasons these crops are facing a lot of problems in the cultivation time. Okay, next the example of cherry ring spot diseases in the pollen grain. So next, insects. And after the seed transmission, the majority of the plant viruses are transmitted through the insects. These insects are generally called vectors. You know vectors?
Yes, sir.
Yes, sir.
So like mosquitoes, aphids, white flies, many. These are the function as the vectors. Most of the mosquitoes, aphids, thrips, and white flies very dangerous in our Indian agriculture system. And any plants, any crop, these problems are going on till now. So, and these nowadays, these aphids, thrips, and white flies and mosquitoes, they are resistant to the chemical pesticides also. So the insect example of disease: rice tungro, potato leaf roll, papaya mosaic. These are the examples of the insects. Nematodes. Nematodes belong to the genera is Zyphonema and Langidorus, known as the. It is involved to the transmission of the soil-borne viral diseases. So this is are very less because our farmers, they are using a lot of pesticides and biopesticides. So that time the soil-borne diseases are killed by these pesticides. And fungi. Some fungi have been reported act as the vector for the some soil-borne viruses. Example, Olpidium brassicae is the noun-transmitted tobacco necrosis virus, and through the juice pores. So next, Phanerogamele parasites. The angiospermic plant parasites Cuscuta was proved the transmitted virus through the Astoria. So body fluids, some animal viruses, like it is related to the body fluids, like human beings. The animal viruses are transmitted through the body fluids, like AIDS case caused by the HIV is transmitted through blood and semen, and also some the virus HB, hepatitis. Okay, these are some blood fluids diseases. Next, how to control of viral diseases. Any idea? Yes.
Self sanitation, sir.
So generally, many control mechanisms of many controls are there, but not all the times some pesticides are not controlled by the viral diseases. So like same like already I told you some of the yes. Okay. So for example, already I told you I recently some some 50 lakhs grant from DBT, Department of Biotechnology, Government of India. So actually we are growing in the every year growing in the chili crop. So in the 2 years back, one thrips attacked to the chili crops, that is black thrips. The scientific name is Thrips parvispinus. So the thrips is resistant to the chemicals, pests. So we are doing in the different ways. We are working in the lot of work in the 1 year. We are using some different pesticides, different chemical pesticides. First of all, we are using different chemical pesticides. There is no results. We are using in the different chemical pesticides in the combination of high dose of pesticides. The thrips, the black thrips not killing. So we are using some, first of all, biopesticides. The thrips is not killing. So we are using in the different technology in the microbial surfactants. So after 1 year, we are using some microbial surfactant. You know microbial surfactants means?
No.
Yes, anyone?
That acts on the surface.
That is the role of surfactants. The microbial surfactants, it is nothing but the suppose we are using biosurfactants. Biosurfactants is the bacteria released by the some metabolites. That metabolites we are using killing the black thrips. So we got some good results. So immediately we are applying the grant. So some 6 months process are there. Some presentation, some evaluation processes. Finally, we got the that grant. But what happening here? The thrips is not originated in India. The thrips origin from the Indonesia. That is migrated from the Indonesia in India. So that is resistant to the chemical pesticides. We are using high doses of chemicals also, but we are not killing that thrips. It is look like a hair-like structure. Hair-like structure means very small, very thin, but the high doses also they are resistant to the chemical. So the nature of the capacity of the thrips. So many ways are there. Some organic ways are there. Some chemical ways are there. Some biopesticides are there. Some different ways are there. So according to our syllabus, some of the general we are using the control of viral disease in the general processes. The general processes is the suppose the insect vector should be eradicated by the spraying of insecticides. Generally, suppose the viral diseases through the insects. So some insecticides are there. Some fungicides are there in the market available. Many drugs are available. Fungicides, insecticides, and the source of infection should be removed. The infected plant debris should be borne. Diseased tubers, bulb, rhizome, corn, stem cuttings should not be used for the cultivation. The weeds harbor viruses should be destroyed. So these are the general thing. And resistant varieties must be cultivated if they are available. Resistant varieties, there are some government agencies and some agriculture units, they are developed the resistant varieties. And the immunization of the plants with mild viruses strains give the protection to the plant from the serve the effect of certain virus strains. So, and also rotation of crop, it is very, very useful. But no, most of the farmers not doing the rotation crop. Suppose this year same plant, next year same plant, next year same plants. What is happening? Without rotation of crop, already the previous bacterial diseases, previous viral diseases in the deposit in the soil only. So, so we are taken different varieties again cultivated in the same plant, immediately attack to the these viral diseases. It is, but the crop rotation is very, very important. And also exclusion of the source of infection. Exclusion of the source of infection. This method preventing the entry of the source of the infection into the area does not naturally exist. So it is strict enforcement of quarantine laws restricted to the entry of the infection. So next, the quarantine means, this word we are popular using in the corona time. Yes, quarantine means
Medication.
Isolating.
Yes,
Isolating, sir.
So prophylactic measure, it is used to prevent the entry of new diseases, insects, weeds from the other countries. Generally, you observe this in airports. Any plant material, any some quarantine department are there. So they are doing some tests developed by some test. When the plant material, yes, quarantine tests done, then the material is delivered to the customer. Okay, so in every international or national airports, the quarantine departments are there. Okay. So in this isolation of the source of infection, the isolation and complete eradication of disease hosts, the common practice preventing the plant disease from the first few plants is field detected with infection should be destroyed by roughing and burning. The wild plants, mostly the weeds, can serve the host, similarly eradicated. So immunization, this method aims to making the host system resistant to the disease by vaccination, by immunization. What is the immunization and vaccination in these plants? Like we are using some plant growth biofertilizers. So eradication of vector. The many viruses are known to the transmitted by the insect vectors. Vector control considered as the important strategy, the preventing the viral diseases. The destruction of the insect vectors could be achieved by the using suitable insecticides. There are many companies are developed the insecticides. The insecticides, it is nothing but some chemical pests. Chemotherapy. Chemotherapy means chemicals like cytovirine and thiouracil 2,4-D, zinc sulfate, malachite green. These are many of these in this chemical. The farmers also, they are using lot of chemotherapy. Chemotherapy means chemical pesticides. They are using chemical pesticides. They are the entire pollution, and nowadays any vegetable crops, any plants. So the farmers spraying some chemical pesticides in the vegetables. Some of the small quantity of the chemical pests deposited into the like tomatoes or whatever any vegetable crops. It is automatically deposited. They are not dissolved. So directly we can buy the market. The market also, before market, the market sellers, they are using, suppose fruits, carbide, they are using in the storage purpose and the ripening purpose. They are using different chemicals they are using, but that dangerous chemicals deposited in the parts of the like vegetables, in the parts of the any fruits, any leafy vegetables, but we are directly by the market, directly we are cooking, but it is very dangerous to the human beings. Okay, because they are in the cultivation process, they are not control all the viruses, because the viruses in the, suppose some environment change, the virus attack viral diseases, some environment, some cold, the bacterial disease change. So that's the reason the farmers, they are using most of the highest percentage of chemicals are using in the cultivated the commercial crops or vegetable crops, whatever may be, but it's very dangerous. So that's why the government are encouraged to the organic food. But the farmer start a organic food, the farmer not getting the more money. So and in the available in the like bio seeds and hybrid seeds are there, but till they are using mostly like for example is chicken is ill. Generally the chicken is developed in the 2 months, but after 25 days they are giving many injections to the chicken, so automatically the chicken is a large 2 kgs 3 kgs. So that's why you difference between the suppose in villages natukodi. So it is some natukodi, you know, natukodi means it is developed in the naturally in the villages. It is take at least 6 months time, but compared to the boiler, within 45 days the boiler chicken, it is more than 2 kgs, more than 3 kgs. It is because of only the injections. So here the farmers are highly involved in the chemical therapy. So that's why the central government is encouraged to the organic. Okay. So next, the breeding of disease resistant varieties. Different methods adopted the viral plant disease control, breeding and cultivation of the resistant varieties seem to be the most effective method. The resistant varieties, the host system becomes immune to the virus infection. So the resistance virus developed is it take long time, but the government developed the resistant virus, but the private companies are developed in easily in the bio and some hybrid. The farmers choose the, it is easily available and easily the more production. In every farmer, they are not care about the in the future generation. So in actually recently I saw the in some villages. So after cultivation of rice fields in Punjab, Haryana, immediately they are burning the rice straws. You know rice straws after cultivation of some harvesting of the paddy crops, then remaining they are burning. So due to this, in Delhi the pollution is very very high. Only due to this, the Punjab farmers and Haryana farmers, they are doing in the burning of the rice straws. Here in remaining states also, remaining state farmers also started the rice burning straws, because there is the some technology developed, some tractors are available, some harvester machines are available. So that's why, and in villages it is reduced the animals like cows, buffaloes. It is reducing the almost all in the 90% of the villages, the very less number of cows and animals. That's why our Telugu states farmers also started in the rice straw burning. So when rice straw burning, so the farmer not thinking in the future generation what happening in the rice burning straws. So the fog, it is polluted in the entire environment, and also the pests is deposited in the rice straws only. When burn the that rice straws, it is the chemicals are polluted in the environment. It is polluted in the, it is harmful to the some useful microorganisms. It is harmful to the birds. It is harmful to the entire biodiversity. So this happening already started in our states. So don't do the this type of rice straw burning, because what happening in the Delhi, the pollution in the coming days, in the after 5-6 years, same happening. Please mute yourself.
Sir, it is Navadeep. Mute that, sir.
Okay.
Okay, this is the some control of viral diseases, right? And also okay, this is the chemotherapy. these are the very some general control of viral diseases, right? So do you understand today class? This is the today class. So tomorrow we'll discuss. Tomorrow we will discuss in isolation and purification of the plant viruses, and as well as we are missing in the bacteria class. We are missing one topic, right? Mycoplasma. You know mycoplasma? Very simple. That is also some important question. So day after tomorrow I will cover that also. What is mycoplasma? That is also important question, long or short. What is the mycoplasma?
These are the smallest microorganisms.
Smallest microorganism. Okay. Okay. So we will discuss day after tomorrow.
0.1 nanometers in size, sir.
Okay. We'll discuss. Very simple. What is the mycoplasma? First, you are saying the answer is not. It is not smallest. It is there. What is the
Pleuropneumonia like organisms, sir. PPL.
No. What is the structure? Cell wall present or not? Lack of cell wall.
Cell wall is not there, sir. They are like bacteria cells, but the cell wall is absent.
Sir, lack of cell wall, sir.
Yes,
it is like a
cell
bacteria.
wall. So
it is a bacteria, but the cell wall is absent. Bacteria cell wall is made up of peptidoglycan, but here the cell wall is absent.
Okay. So we will discuss in tomorrow. So entire actually 90% of the virus chapter is closed. So remaining these two topics: isolation and purification of plant viruses, and remaining mycoplasma. Then we'll go for the next algae. So anyone, please send me the first page of the. Actually, I missed your C1 book. So the syllabus copy, na? The first black 1, black 2, black 3, black 4. Anyone take the one photo, then send me. Bamina is there?
Yes, sir. Yes, sir.
Just
Yes, sir. She's
take one photo. Please send me on the first page on your book. Black 1, black 2, black 3, black 4. So we are
yes, sir.
going to the next classes, algae and fungi, right? So what is the general character of algae?
Fungi 1, fungi 2, sir.
Okay. There is no algae?
No, sir. No algae, sir. Fungi 1, fungi 2.
Okay, just you send me that part.
So in the block 1, we have viruses, bacteria, fungi 1 and fungi 2. I will send that one.
Okay, next block 3.
Block 3 is fungi 1, sir.
Block 4?
Fungi 2. That is masticomycotina and zygomycotina.
Okay,
with ascomycotina and all this stuff.
Block 1 and block 2 is only bacteria and viruses, right?
Yes, sir.
Sir, okay, no problem. Just you send me the picture of this.
I'm doing it right away, sir.
Okay. Thank you. Any doubts?
Sir, the presentation పంపిస్తారా sir?
పంపిస్తాను.
Okay, sir.
Right. Thank you.
Okay, sir. Thank you, sir.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
So do you want me to send it personally or in the group, sir?
No, personally. My WhatsApp number is there, na?
Yeah, I do have.
Yes. Everyone know. Everyone have books, na?
Yes, sir. All have.
Yes, we have.
Yes. Only except me. Yes. Thank you.
Thank you, sir.
Thank you, sir.
So just tell